Wonderful. So thank you everyone for coming and for joining our webinar. We're just going to wait until it's about 2 o'clock Pacific time, 2:01, 2:02 for people to join, and then we will start with the webinar. Okay, I think we are going to start the webinar. I just want to introduce myself. My name is Amaka Obidegwu, and I am the Project Research Manager of the Army of Women. Thank you everyone for joining us. And if you have questions throughout in the webinar, you can type them in, and then we can address them at the end when we have our question and answer session with Dr. Spencer. And so here joining me, I also have the legendary Dr. Susan Love, and she will be facilitating a lot of the conversation and the questions at the end, and then also we'll be able to provide some tidbits as we move along with this webinar. And just as an introduction, our researcher who will be presenting today is Dr. Juliet Spencer. And she is a professor of biology at the University of San Francisco. She received her PhD in microbiology from the University of Virginia and was a Parker B. Francis postdoctoral fellow at the Carter Immunology Center. She worked as a drug discovery scientist for ovarian cancer at Ceratec LLC for 3 years before joining the faculty at USF. Her lab focuses on the herpes virus and understanding how these viruses manipulate the human immune system, and more recently on how these viruses may contribute to chronic disease. Her work has been recognized by USF with the Distinguished Research Award and the Dean Scholar Award, and she has also been recognized by the Association of Women in Science for her work mentoring female students. We're so happy and glad to have her today to talk about her current study that she has with the Army of Women. She'll be discussing the study in more detail so that you all can know about this important research that she's doing and hopefully be able to participate or refer friends to participate in this very important study. So Juliet, I will pass it on to you.
Great. Thank you so much for the introduction. And I just want to say I'm so honored to be invited to speak with you today, and so grateful to Dr. Love and the Foundation and really the entire Army of Women for this opportunity to tell you about the work happening in my lab at USF. And so our goal is very simple. We want to develop a test for the early detection of breast cancer, and that's because early detection saves lives. The earlier you detect a developing tumor, the better the outcome for the patient. So the current study that we are running has 3 very specific sub-goals, and they're shown here. To develop a test to detect viral IL-10 in blood, to enroll 300 women for a case control study, and then to screen the samples that these women generously provide for viral IL-10 and other immune markers to determine a signature profile for breast cancer. And the study is currently in progress, so unfortunately I can't tell you the outcome today, but what I will tell you is some of the background research that led us to this point, and also share with you what progress we have made so far on these goals. So at this point, you might be wondering, what is viral IL-10? So let me take a moment to explain and give a little bit of background on that. So viral IL-10 is a protein that we're exploring as a potential biomarker. It's a protein that's made by cells that are infected with cytomegalovirus, and it's a protein that has really potent immune suppressive properties. And so how much of that protein is produced may reflect other things happening in the body, such as a particular disease state or specifically the development of tumors. So let me just say a few words about biomarkers. So the National Cancer Institute, which is one of the institutes for the NIH, or National Institutes of Health, defines a biomarker as a biological molecule found in blood or other body fluids or tissues that is a sign of a normal or abnormal process or of a condition or disease. And biomarkers are really important, and they have 3 primary uses. They can be used to diagnose a condition like early stage cancer, and that's what we're very interested in. They can also forecast how aggressive a condition is, so they can help get a picture of the prognosis or the likely outcome. You know, where does this particular condition fall in the range of expected outcomes. And then lastly, they are used, and they're very important for predicting how well a particular patient will respond to therapy. And so for breast cancer, the main biomarkers that we have right now are things like ER, which is the estrogen receptor, or PR, progesterone receptor, and HER2, and that's a specific type of epidermal growth factor receptor. And knowing whether your tumor has any of these biomarkers will inform the course of treatment that your physician selects. And so, for instance, patients who have HER2 may receive Herceptin, and that's because it's a drug that specifically targets the HER2 receptor, whereas this treatment is not likely to be of much benefit for women who don't have HER2. So I mentioned that we're exploring viral IL-10 from human cytomegalovirus as a potential biomarker. And if you've never heard of cytomegalovirus, you're definitely not the only one. Most people have never heard of it, which is funny because most people have it. So here I have a list of potential risk factors for breast cancer, and really Dr. Love wrote the book on this, but just to give you a few highlights. So there are some risk factors, such things that you really can't control, like your family history, and in particular, whether you have certain mutations that might predispose you. So these are genetic factors like BRCA or BRCA1 and BRCA2 mutations. And I have a picture here of Angelina Jolie, who has famously shared her experience finding out about having these mutations and had a double mastectomy proactively to sort of avoid the possibility of developing cancer. And so I did want to take a moment here to distinguish between genetic markers and biomarkers. So genetic markers reflect potential. So they are an indication of a particular sequence in your DNA that conveys a higher chance or a greater risk that cancer may occur in the future. So genetic markers reflect potential risk. Biomarkers are a readout of the current state of the body, and so biomarkers are useful for informing the actual diagnosis and treatment. But these are things, as I said, these are genetic markers that may contribute to higher risk. There are also some issues surrounding pregnancy that contribute to higher risk, such as not having children at all, or if you do have children, actually that can lower your risk unless you have children past the age of 30. And then the other factor that's important, or can be important, is high breast density. And right now, California is one of about 35 states that require women to be notified if they have high breast density. So you might receive a letter in the mail after your mammogram informing you that you have high breast density. And one reason this is considered a risk factor is because with the tissue being more dense, it's more difficult for the mammography to pick up small developing tumors. So that's one reason why, for the risk with the breast density. And I do want to emphasize, however, though, that mammography is currently the best tool that we have to detect breast cancer. On the other side, I have some lifestyle factors, and these are things that you might have a little more control over, like whether you have used or you use or have used menopausal hormone therapy or hormone replacement therapy, as well as things like your weight, your level of physical activity, use of alcohol or tobacco. One thing that's recently been added to this list is working the night shift, and we're still understanding the specific reasons for this, but it seems to be related to disruption of normal biorhythms, and in particular, the increased exposure to light at times outside of the normal daylight hours, which tends to decrease secretion of melatonin. And melatonin is a hormone that's secreted by the pineal gland in response to decreased light. So normally, you should have high levels of melatonin at night, and this plays an important role in regulating other hormones, especially things like estrogen. And so the night shift tends to disrupt that regulatory sequence, and so that may be another reason why women who regularly work a night shift tend to have a higher incidence of breast cancer. But back to my study, I told you that we wanted to use a protein from cytomegalovirus as a brand new biomarker, but you'll see that cytomegalovirus isn't on this list. So in fact, there is no infectious agent bacteria or viral on this list. So what about the connection between infectious agents and cancer? Well, right now, we haven't found any really strong links between a particular infectious agent and breast cancer. But I will say we're learning more every day, and it really wasn't that long ago that we first discovered that cervical cancer is specifically caused by infection with certain strains of human papillomavirus, or HPV. And it's been a relatively short span of time that we've gone from understanding that connection to having a vaccine to prevent cervical cancer. So wouldn't it be amazing if we one day have a vaccine against breast cancer? I think that's a dream or a goal we can all hope for. But in the meantime, what we can do is, here I have a little inspirational quote from Gandhi, and the future depends on what you do today. And so this is why I think it's so important to promote research to explore every possibility, because we just don't know what will lead to a major advance until we do the work and do the investigation. So now I'm going to really focus on cytomegalovirus, which is what we focus on in my lab, and it's in the herpes virus family. So here I have a list of all the herpes viruses that infect humans, and probably most of us are infected with at least one or more of these viruses. Now, you may be familiar with herpes simplex type 1, and that causes cold sores, or you might be familiar with varicella-zoster virus, which causes chickenpox. And one thing common to all these viruses is that once you're infected for life. And that's why some people get outbreaks of cold sores that recur. They periodically, every time they get stressed or they get a sunburn, they get another outbreak of cold sores. Or why people who had the chickenpox as kids can develop shingles later in life. So I've got my cytomegalovirus down here, and it's one that most people have never heard of because it doesn't cause a really noticeable rash most of the time during the primary infection or during the recurrence. But it can be a problem for certain people. And let me just go forward here. Let's see, I had a cartoon picture of the virus on this slide, but here I have an actual picture of the virus. This is herpes simplex virus, and it's a picture taken from an electron microscope. And these are just some common features of the herpes virus family. The structure, which is shown here, so they have this sort of this capsid with these little projections that you can see, and they're surrounded by an envelope which it steals from a host cell. They're also very widespread in the population. As I mentioned, most people have been exposed and probably have at least one or more herpes viruses in their system at any given time. And then last is the ability to establish lifelong latent infection. And so what that means is that the immune system can never really completely eliminate the virus the way it can for other things like influenza or viruses that cause the common cold. So that's why I say herpes is forever. And I don't know if this is helpful, but this is how I tend to think about interactions between herpes viruses and humans, is a bit like a fencing match where the virus attacks the host, and this is the first time you've been exposed, and the virus is trying to infect cells because that's the only way it can make more of itself. And then the idea is that your immune system is very good at recognizing foreign invaders, and the immune system for most viruses, like influenza, even though you might not feel well for a week or two, the immune system is very effective at launching a counterattack and eliminating the virus. And so it's really just this herpes virus family that has become so well adapted to living with humans that herpes viruses actually aggressively launch a counterattack. And so instead of just laying down and taking it and getting eliminated from the body, herpes viruses will actually actively manipulate the immune system, and they will prevent the immune system from being able to eliminate them from the body. What ends up happening for most people who are otherwise healthy is that there's a sort of delicate truce, and the virus and host can sort of peacefully coexist. And the virus is dormant most of the time, unless there's some sort of imbalance in the body due to stress or immune suppression or any number of other things. And so this is one reason, since virus reactivation is a good readout for an imbalance in the body, this is one reason why we're interested in viral IL-10 as an indicator for the development of breast cancer. So one thing people ask me a lot is how might you get cytomegalovirus? And the answer is simple. It's usually through a smooch or something from a well-meaning relative. So the transmission is at mucosal surfaces, so that means the virus is shed in things like saliva. And so it's just very casual contact that can transmit the virus easily. And so most people are exposed at a very young age, usually in preschool. This is actually a picture of my son, Dimitri, when he was in kindergarten, and I was invited to the classroom to give a little talk about hand washing to kindergartners, and I can tell you that you don't have to spend much time in a kindergarten classroom to see exactly how this virus is so easily transmitted among people at a young age. And so that's why by the time people reach adulthood, the vast majority of people are seropositive, meaning they have evidence of having been exposed to the virus. And the first exposure, the infection that occurs for most people when they're in preschool or kindergarten, is very mild. Often there's no symptoms at all, and so that's one reason why people aren't necessarily aware of it. As I said, cytomegalovirus is one of these herpes viruses that can establish lifelong latency. And for cytomegalovirus, the site of latency is in progenitor cells in the bone marrow. So that's where it will have a reservoir, and it's in the body forever, but the virus can undergo sort of low-level periodic reactivation and shedding, and that shedding occurs in saliva, or it can be in urine, any type of body fluid, and that's another way that it's very easily transmitted amongst the general population. So most people will live peacefully with cytomegalovirus most of their life without any complications. I showed you a few slides ago. The folks that do have issues are women who are pregnant. If they get a primary infection, it can be problematic for the developing fetus. And fortunately, that doesn't happen all that often, although it does happen. And the other population that has to worry a little bit about cytomegalovirus or CMV are people that are undergoing transplants, and that's because they have to have immune suppression so that they won't reject the organ. And then, of course, this creates an environment where the virus is unchecked and it can really replicate to high levels and cause problems. But for the average person, you'd probably never even know that you have cytomegalovirus and never have any problems with it. At least that's what we think. There's been a growing interest in the field lately trying to understand whether cytomegalovirus might contribute to sort of long-term diseases that are prevalent in our population and we might not associate with cytomegalovirus or with any infectious agent, and that's things like cardiovascular disease and particularly cancer. And so one idea is that the virus, the long-term chronic infection with a virus like cytomegalovirus, which is very good at sort of modulating cell functions, coupled with declining immune function, which is something that unfortunately happens to all of us as we age, our immune system just gets a little bit less robust, can combine and lead to cancer. And I think that most of the folks in the field would not suggest that cytomegalovirus causes cancer per se. I don't believe that's true, but I do believe it can be another factor. It can be something that can contribute or exacerbate a situation where a tumor might be inclined to develop anyway. So sometimes we use the word oncomodulation, and that just means the ability to modify or tip the balance in favor of tumor formation. That's, I think, where most people are thinking, most people in the field are thinking of CMV or cytomegalovirus right now. In terms of the connection between cytomegalovirus and breast cancer, I'll tell you the truth, the jury is still out on that. There are some very nice studies that have been done that seem to very strongly support the notion that CMV is linked to breast cancer. And here I'm just showing you one citation. This is work from Dr. Cecilia Soderberg-Norrklar in Sweden. And her group has been at the forefront of this effort. She has found, she has access to a number of specimens from in Sweden, and she has found evidence of cytomegalovirus in basically all the samples of breast cancer that she has examined, and these are tissue biopsy samples. There are some other groups.
Wait, can I butt in just a second? Did she
yes,
look
please.
at, this is Dr. Love, did she look at normal breast tissue and normal lymph nodes?
Yes, I believe so, and I think this is one figure that I've swiped from her her paper, and so she does have some matched normal tissue. The lymph nodes, that I'm not so sure, but actually the paper here, she had some sentinel lymph nodes, so I believe she did have node tissue as well. And that is where the confusion lies, because some people don't find CMV in the breast tumor samples, and some people do find CMV in the normal tissue. So Dr. Soderberg-Norrklar's lab they say they just have really good technique. Other labs say, well, we don't agree, we don't know, that's not what we see. So that's why I say there's definitely some debate in the field right now as to the real role of cytomegalovirus in cancer. So that's a really, really great point. And obviously, I just told you that the vast majority of us walking around have cytomegalovirus, and yet we know we don't all have cancer. And so that's a really great point. And I think that's why I would argue I do not think that CMV is a cause of cancer. I think it can be a contributing factor. So thank you. That's a really great point. So as I say, the jury is still out. Now, this is a picture that I'm still trying to find a better picture. I know it's very confusing and overwhelming, but what it shows and what I want to tell you is this is the part where how I kind of got involved in the field of looking at CMV and breast cancer, and that's because what I'm interested in is how the virus manipulates the immune system. And I just want to take a moment to explain about how the immune system works and why it's a really different system than a lot of our other systems in the body. So the example I like to use is the respiratory system. And, you know, the respiratory system, the lungs, it's very simple. We breathe in, we breathe out. Everybody is on the same page. Or even the heart, the circulatory system is a great example. All the cells in the heart are connected to one another. And so it's basically at the same time, beat, rest, beat, rest, beat, rest. Everybody's on the same page. The immune system is a little bit different. And the way it works is that these cells are scattered throughout your body, and their function is surveillance. They're looking out for something that doesn't belong. And the problem is when they find it, they need to be able to communicate that to other cells and sort of, you know, get the troops mobilized and respond. And unfortunately, the cells in our body, and that's what's being depicted here, all these little circles are different types of immune cells. So unfortunately, the cells in our body don't have cell phones, so they can't, you know, get texts or tweets or anything like that. But what they do is they release soluble proteins, small molecules, into the body fluids, the blood and the lymph. And the cells that have the right receptor can receive the signal. So it's a little bit like following somebody on Twitter to be open to that channel to be able to receive the information. And so I like to show this picture of how complicated our immune system is because cytomegalovirus is actually a pretty big virus. It's got 167 genes. And I know that that might not seem large compared to the human genome, which is about 30,000, but 167 genes for a virus is huge. And just to give you perspective, HIV has 9 genes, and we still have not made much progress in stopping this virus. So cytomegalovirus has 167 genes, and to me the most remarkable part is that more than half of those genes have nothing to do with virus replication at all. They specifically target host processes, and most of them are aimed at manipulating the immune response. And what we're interested in my lab is understanding how cytomegalovirus manipulates this communications network and how it can disrupt the flow of information between immune cells, and that's in part what leads to the failure of the body to eliminate this virus and what enables the virus to sort of peacefully coexist. So in my lab, we are really interested in one particular immune communicator. And these immune communicators from this previous slide, you can see all these names, IL-4, IL-5, there's about 20 or 30 of these ILs or interleukins. And most of these guys have the function of inflammatory responses, and that's great because what that means is when an immune cell senses a foreign invader, what it can do is send out the signal, and what we want, in this case, we want inflammation because inflammation is going to bring cells to the site, it's going to bring fluids to the site, and it's going to give an opportunity for the immune system to respond. And that's what most of these cytokines do. They are inflammatory. And one of the things I learned studying cytomegalovirus is that, of course, inflammation is not always a good thing. And so in particular, there's a cytokine, an immune mediator known as interleukin-10. And this is a normal part. Everybody has it, and it's actually quite important because when you do have the recognition of something foreign in your body and you have this inflammatory response, you need a way to end it. Chronic inflammation is very bad and destructive. And so interleukin-10 has a really critical role in ending the immune response and saying, okay guys, great job, we've cleared the virus, everybody can go home and resume your normal functions, we really don't need any more of this inflammation. So interleukin-10 is an anti-inflammatory cytokine, and it's really critical for getting the body back after an immune response. The problem is that these herpes viruses, they have just evolved with us so exquisitely that the virus said, you know what, that would be a pretty cool tool for me to have right at the onset of my infection. So the viral version, this is a version of this human gene that has been captured by the virus, and it mimics the effects of human IL-10. And the biggest thing that it does is the virus can make it immediately upon infection, and it shuts off the immune response. And instead of the immune system mounting a large response to clear the cytomegalovirus, instead it gets the signal from the viral version that, oh, show's over, there's no problem here, everybody move along. So the viral version also has anti-inflammatory effects. And that's a pretty cool tool for a virus to have, particularly a virus like cytomegalovirus that wants to hang around for a long time. The problem is, do either of these guys, the normal interleukin or the viral interleukin-10, have a role in cancer? And I think this is where I can try to explain a little bit how maybe some of the observations of some of my colleagues, where some people see virus in cancer, some people don't see virus in cancer, some people see virus in normal tissues. I think that this might help explain a little bit of how that all works. And I'll just, actually I just want to mention some literature. The thing that got me started down this road was finding some papers in the literature suggesting that sometimes the human cytokine itself, the human IL-10, is found in very high levels in women with cancer. And that's in the blood. And there have been some studies suggesting that human IL-10 can also, the normal human cytokine, can promote tumor progression. And then we just put a few other pieces of this puzzle together. One report that IgG, or immune response to CMV, has been noted to get higher before breast cancer, and then some people finding evidence of CMV, and that was the paper I showed you earlier. That's the Taylor et al., and there have been some other studies confirming this. And so I don't know entirely, but I think CMV IL-10 could be a link between a lot of these observations. And so this is the schematic that I have. And so over here I have a cell that's infected with virus. And so the idea is this cell has been infected and it's making new virus particles, but it's also producing these little blue blobs are meant to represent the viral IL-10. And so the idea is that viral IL-10 can be released from an infected cell, and it can act on any cell in your body, infected or not, as long as that cell expresses the IL-10 receptor. So as long as that cell has the mechanism to receive the normal IL-10 signal, it can receive signals from the viral IL-10 as well. So this is where I think whether you find the virus or you don't find the virus, maybe that's less important than whether you find the viral cytokine in the tissue. And so the specific question that I was investigating, and this is kind of what led us to the study that we're embarking on now, is can viral IL-10 act on breast cancer cells to promote a more invasive phenotype? And I have this term paracrine manner, and paracrine means usually it's acting a relatively short distance. So it's not produced by the same cell, that would be an autocrine effect, and it's not produced by cells far, far away in the body, that would be an endocrine effect, but paracrine. So can there be virus-infected cells in or near the tissue that release the cytokine and may change the behavior of cells that are already predisposed or already leaning toward a potentially cancerous phenotype? And so the first thing we did to investigate whether this theory was even possible was to find out whether breast cancer cells express the IL-10 receptor, because without that, then they would not be likely to receive the signal. Now, so this is work that was done in my lab with using breast cancer cells in culture. And I just want to share with you, if you haven't read the literature that uses breast cancer cells in culture as a model, if you do look at it, what you would find is that the vast majority of studies use one of these two cell lines. About 70 to 80% of research studies published using breast cancer cells in culture use either this MDA, MB231 cell line, or this MCF-7 cell line. And so something we're very interested in always with these cell lines is what type of biomarkers they express. So the MDA cells are from a patient that had a tumor that was considered to be triple negative. So it's negative for estrogen receptor and progesterone receptor, which isn't shown here. And it's also negative for HER2. The MCF7 cell line is very widely used by researchers, particularly those interested in cancers that are estrogen-driven, because it does, and this is from a patient who had an ER-positive tumor. So it does, the MCF7 line does express the estrogen receptor, and neither of these two, as I mentioned, express HER2. Actually HER2 is a relatively small percent of the total breast tumors that are diagnosed express HER2. But what we found is that both of these cell types do have the IL-10 receptor. And this is just shown here where I have this is a picture if you've never seen a breast cancer cell in culture. Most of them tend to have this very long, stretched out appearance. And then the blue is the nucleus of the cell. That's where the DNA is. And then the red speckles, that represents staining for the IL-10 receptor. And so these are cells. This is the MDA cells, but the MCF7 cells look just like it. And they have plenty of IL-10 receptor on their surface. And this is the visualization of that, and then this is just a measure that we use, flow cytometry, to get a quantitation of how much fluorescent stain the cells are binding to. And so yes?
No, a quick question. This is Susan Love again. Do normal breast cells express the receptor?
Yes, they do. A lot, in fact, most cell types in the body, actually, almost all cell types express the IL-10 receptor. A lot of cell types do. So again, this is where I would say I don't think that just being exposed to IL-10 or viral IL-10 is the only thing that can contribute, but I do think that a cell that is leaning in this direction can be pushed over the edge by the viral IL-10, but a lot of cell types express IL-10 receptor. Yes, great question. Thanks. Okay, and so, but what we found is working with these breast cancer cell types, so the MDA cells are really, now I just want to emphasize, the MDA cells are considered a very aggressive form. So what we found is that when we cultured these cells in the presence of viral IL-10, they grew even faster. I mean, they grow great in culture anyway. These are cancer cells. You can do a lot of things to them, and it's kind of hard to kill them. But one thing we found is that if we expose them to viral IL-10, they actually grew even faster, which is crazy. And if we try to if we treat the cells with chemotherapeutic agents, and somehow I must have lost my label here, but these down in this lower panel, these are cells that were treated with etoposide, which is an inhibitor that sometimes is effective at inducing damage and preventing tumor growth. We see that the normal tumor cells they're stopping growth, they're starting to die, so they were responding to the etoposide. But cells cultivated in the presence of the viral IL-10 just keep on growing and growing and growing. So cells that are exposed to viral IL-10 grow faster. They can survive in the face of drugs and other signals that tell them to try to kill them. And they also exhibit increased movement. And this is over here, this is a measure of cell migration. This is a kind of a tricky experiment, but the way we measure movement and particularly tissue invasion in the lab is we use a chamber where we put the cells in the top and we put some sort of stimulus in the bottom, and you can see that our stimulus is called EGF, or epidermal growth factor receptor. And the cells really like it, and they want to move toward it. So they have to move through the pores of this filter and make it into the lower chamber. And when we include the viral IL-10, what happens is we get even more cell movement. And one thing that we do in the lab to simulate invasion is we can coat the pores in these chambers with a substance called Matrigel, and Matrigel simulates the normal basement membrane that you might find in the body, and so cells that are exposed to viral IL-10 are very good at busting through this. And so what does all of this mean? Well, here is a picture of a developing breast tumor. And so, you know, in a perfect world, this would be cells that are starting to grow out of control, and they're forming a small tumor mass. And this would be maybe what we would call DCIS, or ductal carcinoma in situ. So this would be a very localized lesion. And so here I've got a picture showing you.
Wait, ductal carcinoma in situ doesn't give you a mass.
Okay. So
it's
right, so
the duct.
it would be a small, tiny clump,
Invasive?
say.
Okay.
No.
Sorry. You can't feel a ductal.
Right.
I mean, you're not going to have anything to feel because it's within the milk duct.
Right. So it's going to be very, my point was just that it's going to be very small and localized. Okay. And sorry if I misspoke or made that sound incorrect, but the point that I was trying to make is just that it's very localized. And so the problem is here, if we consider the developing cluster of cells here, the primary tumor site, that as the cells begin to grow more and they begin to resist signals for cell death, and they begin to become more migratory, what they can do is, this is when they can bust out and they can invade the surrounding tissue. And so that here, this red line, that sort of represents what I was describing earlier in my experiments where we use Matrigel. That's meant to represent sort of the tissue borders where the tumor cells would begin to invade surrounding tumors. And then the sort of worst case scenario would be where the tumor cells actually begin to enter the bloodstream, and they would make it into the general circulation. And those tumor cells, these circulating tumor cells, may then be able to find other hospitable sites in the body that enable them to form metastatic lesions or secondary tumor sites. Now, in the case of breast cancer, the first thing that happens, and I like this picture a lot because these sort of thin lines represent the lymphatic system, and so these sort of dark dots represent the lymph nodes. And so all the fluids that drain from the breast tissue will first go through these lymphatic ducts and into the lymph nodes. And so the lymph nodes are, of course, the primary site where we might see tumor cells away from the primary breast tissue in breast cancer. And then, as I say, the worst case scenario, I believe, would be if you have those cells then being really aggressively invasive and making their way into the circulation, and that would enable them to enter other sites, and some of the more common sites are the liver, the lung, and it's not shown here, but also the bone marrow. Okay, so this is what we would really like to avoid. And so our concern is that what we've seen is that viral IL-10 has the ability to increase properties that may lead to greater invasiveness, and that could promote the ability of these tumor cells to break away from the primary site and enter the circulation. And so this has led us to ask whether viral IL-10, are their levels high enough that this can be detected in human blood, and do they correlate with disease severity? So this is what we're working on right now. And so this is what kind of gave rise to the Avon breast cancer study at USF, and we are supported by the Avon Foundation. We're very grateful for their funding. And as I mentioned earlier, our first goal was to develop a blood test for viral IL-10. And so I'm happy to report today that we have done that successfully. We have a great test that's very sensitive and very specific. And you may have heard of the term liquid biopsy, and that's a sort of new term that, I don't know how new it is, but it's definitely a hot topic right now. It's something that people are very interested in as a way to gauge what's happening with a tumor or what's going on with cancer that is less invasive than a traditional tissue biopsy that requires, you know, going in and taking out a piece of tissue. So a liquid biopsy is where you use a more minimally invasive technique like a standard blood draw, or even looking at other body fluids like saliva or urine, to detect biomarkers or signs of the tumor. And so in our case, we're looking at detection of viral IL-10 and other immune markers. And other people are looking for things like circulating tumor cells and cell-free tumor DNA. And we are also working to partner with other groups studying these to try and combine and come up with, as I mentioned, a really predictive signature profile for breast cancer. So our first goal of developing a blood test, we've been successful at that, and we are now working on our second goal, which is recruitment. And I mentioned we're trying to get a total enrollment of 300 women, and that is 150 women with a recent breast cancer diagnosis. And we started advertising with the Army of Women in mid-September, and we asked for women with a recent diagnosis that would be interested in helping us. And I want to say a huge thank you to the Army of Women. The response has been phenomenal, and so far as of today we've had 127 women who have responded to our call, so that's about 85% of our goal. And it just takes a little bit of time to schedule these women to come in, and we really appreciate how flexible they are. They're balancing all of their things, their normal life and family and jobs, as well as cancer treatments and doctor's visits, and so it takes a little bit of time to find a great time that they can come in and meet with us, but we've had already 43 women come in, so the response has just been phenomenal. So I would like to say just a big, huge thank you so much, and we really appreciate the Army of Women support and all of you for helping, and we still need volunteers. We're getting close to meeting our goal. But we're still recruiting, and so I would hope if you are interested in participating, or if you know somebody who might be a great candidate, please send them to our website or ask them to email me. We would love to hear from you. And so just a reminder, what we're specifically looking for is women aged 18 to 80 that have a recent breast cancer diagnosis, and the only caveat is that you do need to be in the San Francisco Bay Area to come in for the blood draw, and we'd like to talk to you and get some medical history as well. We have a questionnaire. In terms of the study itself, I mentioned that we're doing the screening, and that is in progress. We screen the samples and process them as we receive them. So as we're getting the samples, we're doing the testing. So we've been doing that all for the past few months, and our goal is to complete the recruitment and testing early in the new year. Then it's going to take a little bit of time to crunch the numbers and do the data analysis, but we expect to have the final results certainly by this summer. We've already got some pretty promising data. I don't want to show you yet because it's preliminary, but we can already see we're already making plans for the next steps, which we believe will be to identify a smaller subset of the women who have graciously participated and invite them to participate in a smaller longitudinal study. And what that means is with this first study, we've been just looking at a single snapshot in time of viral IL-10 levels, and that's helpful for determining whether there is going to be utility for viral IL-10 and the associated markers as a potential signature of breast cancer. But what would be really helpful would be able to monitor those levels over time to understand how constant they are or how they change with disease state. And then one of our other goals is to see if we can make this monitoring as less invasive as possible, and so we are also expanding our studies to look at urine and saliva as well. And so this has been a coordinated effort of many talented students and scientists in my lab, and I really appreciate all of their work, and I'm very grateful for all of the funding from the Avon Foundation, from the NIH, Willie Drake Cancer Fund, Faculty Development, Fletcher Jones Foundation, and very grateful to all of you in the Army of Women for listening and participating, and to Dr. Susan Love for all of her tremendous works. And so with that, I'll just finish, and I welcome any questions.
So I have a question to start.
Okay.
which is, if you want this to be a blood test for early diagnosis, wouldn't you want to get earlier? Wouldn't you want to try people with DCIS maybe?
Yeah.
And have a blood test. Maybe you could have a blood test right before they have their surgery or something like that, and then see if you can use this as a predictor for that rather than later. And then my other thought is, which you didn't mention in your fluids, is you can get both nipple aspirate fluid and ductal fluid from the breast, and it might be very interesting to see whether you could find it in those fluids.
Right. That's an excellent question. So with regard to the first one about using the test as an earlier predictor, yeah, we're really excited because one thing is we deliberately left sort of a vague thing saying we were interested in women with a cancer diagnosis within the past 5 years. So we are getting women at a variety of stages, so that's very helpful for us. And yes, our goal would be to have this be my personal goal is to have this be something that is used truthfully more like the pap smear, that it would be something that women could have as part of their annual screen to monitor levels over time. And that way, when you do see a change in these levels, then I think it would be very useful as an early predictor. That is the way I see this happening, the way I hope that this would work out.
But then you need to get earlier in your samples, because if you just do people who, even though there are different stages of breast cancer, they've already got breast cancer. So if you really want to, what you want to do maybe is have high-risk women, maybe women who have gene for breast cancer but don't have it, or people who have atypical hyperplasia and are being followed, and then some of them will go on and get DCIS or LCIS, and you could follow them. Then you have a predictor. My only worry, it sounds really great, and I think it's a very interesting hypothesis. I just worry that you're doing it in people who already have cancer, and that's not going to serve you in order to find an early detection test.
Yeah, I agree. I think that's an excellent point. And so, originally, so this is part of how it takes a long time, as you know, from the conception to the actual planning and making something happen. And so our original indications were that this might not necessarily be a great early predictor, but it would be something that would be a predictor of the spread of the invasiveness. And that's why we were initially looking for women with cancer. So that was the initial design of the study. And so that was our rationale. However, you are 100% right. And so now that we're a little bit further into it and we're seeing our data, we absolutely see that we need to expand the target groups. And so, yes, we're very interested in starting to identify high-risk women and women who have not yet developed but are in those risk groups, and to find out if this is going to truly be an indicator of the onset rather than the indicator of progression, which is what we originally thought. So yes, we are very keen to work on that, and also very keen to work on other fluids. And so something that I've been telling my lab, some of my graduate students and folks have been seeing a lot of me because this has been a really wonderful time, and one of the things we've been doing is reaching out to a lot of different physicians in the Bay Area and a lot of other biorepositories and tissue banks, and we've been working on getting specimens and samples from all different stages and different types of things. And so it turns out there's quite a lot of paperwork, as you are probably well aware. And so I've been writing a lot of protocols to try and procure many other samples as well, and that is something we're actively interested in. So fantastic.
There is a bank of ductal fluid in high-risk women that is currently at, that was from a previous study, but where there is some 10-year follow-up. So we may be able to at least tell you where that is.
Great. Thank you.
We have a couple of questions from the audience as well. One is,
wonderful.
when are you starting a phase 3 clinical trials on this molecule?
Well, we're not there yet. So I think it's going to be a couple of years. As I mentioned, we do have some additional work to do, and particularly it is going to involve recruiting some of these groups that you mentioned and looking at some of these other fluids. So I think it might be a couple of years down the line, but I mean we're moving forward, and we have a lot of momentum right now. We have had a tremendous amount of support, so I can't give you a date, but I hope it will be soon.
Okay, and the other question from Hari Marsden is, what are the exclusion criteria of your current study?
So right now, the only real exclusion, well, we are specifically asking, as I mentioned, for women with a recent breast cancer diagnosis, and so we define that as within the past 5 years. And the only real exclusion that we have is women who have severe lymphedema who have difficulty with blood draws. So if you have had a double mastectomy, obviously that is not a problem for us unless you have a history of very severe lymphedema and problems with the blood draw. But that's the only exclusion criteria, that and the recent diagnosis.
Okay, any other questions? Come on, people out there. This is your chance. If you understood it perfectly, then I want then you can go to work for their lab. Because
I know, yes, that's right. We need all the hands we can get.
Here we go. Here's one. Will the recorded session be available? I was a little late joining from Brenda Smith. Yes, it will be. We'll post it.
Yeah, and also everyone who signed up for the webinar will get an email in the next 2 days that will give them a link where they can view the webinar and listen in.
Okay, here's some more questions. Bethany wants to know, is there any way for people outside of the San Francisco Bay Area to provide samples without coming to San Francisco?
Yeah, we're working on that now. We had hoped that we would be able to meet our goal within the Bay Area because that increases our pool of people for follow-up studies. But we are just now starting to follow up with people. We have had some fantastic folks who are very enthusiastic about helping, and we really appreciate that. So yeah, people who are outside of the area, they may contact us, and we can try to make arrangements with them to have their blood drawn locally and shipped to us. So we're just starting that process now, so they can contact us if they fall into that group, and we will try and work with them.
Okay, good. Let's see. I would be willing also, but I'm in the LA area, so I did not sign up originally, said Brenda Smith. So yeah, I think that would be good. I know some studies have used some of the commercial blood drawing groups to draw the blood for them as well.
Right.
So you might
exactly.
look at that.
Yeah, and that's more, as I mentioned, this avalanche of paperwork I've been working on with the protocols to get other sites to draw for us, and so that's definitely something we're pursuing right now. So.
Good. Any other questions, guys? You all understood it perfectly, everybody out there? You can, she'll give you a job if you have.
That's right. We need all the help we can get. So I do welcome any questions. I apologize if it was too much virus and if I lost you there, but please feel free to email me with questions.
Well, the interesting thing to me is that it seems like it's not so much that a virus is causing it, but that a virus might be, subverting your way to deal with it.
Yes.
So I think that is, and it may go with a lot of the data about how periods of stress or periods when your immune system's not up to snuff are times when people tend to get more diagnosed.
Yeah, exactly. So obviously, as you well know, there's a lot of factors that can contribute, and we just think this might be one piece of the puzzle that could be important.
Right. And could you give a website again for volunteers?
Yes, that's right. It's spencerlab.org. So SPENCER lab.org. Or you can email me, jspencer@usfca.edu. The website and my email are in the presentation, which I think Amaka said will be available. Yeah.
Yep.
But please, please reach out and contact us. We would love to hear from you.
And we also would encourage people who are signing up to sign up through our Army of Women page, and that will also be in the update email that you'll receive after the webinar is done in the next 2 days.
Yeah, we like to track how many people we actually can deliver.
Yeah, and you're doing a fantastic job. I'm really so grateful for the Army of Women and for the work of the foundation, because it's just a tremendous resource for for people doing research out there to find women willing and interested in and able to participate. It's fantastic.
So one other question, while this is you're doing this research on breast cancer, is there any reason to think that this marker might not be a marker of other cancers as well in the blood?
That's a great question, and you are absolutely right. I mean, we initially investigated breast cancer because of some of the other folks I mentioned in the field, Cecilia Soderbergner-Claire, finding evidence. However, there are also some researchers out there who have found evidence linking cytomegalovirus and potentially viral IL-10 to other forms of cancer. The biggest one where there's the most progress right now is glioblastoma. Yes, and so I would say, those are the two where there seems to be a little bit more than just a flighty connection. There seems to be mounting evidence. So glioblastoma is the other one, and there are a number of groups working on that as well, and we are also working with them to procure samples. We have had a very small subset of samples to test and had very promising results actually on the glioblastoma, but it's a much smaller sample size. So,
and I should say to our breast cancer people, glioblastoma is a brain tumor.
Yeah, it's very poor prognosis. Yes, very aggressive.
Very aggressive one. Yeah. Very good. Okay,
Great.
You get another minute to put your last-minute questions before we thank Dr. Spencer for giving us her time and explaining this to us. And if you come up with questions afterwards, just email the Army of Women, and we'll be happy to forward them along and try to get them answered for you. It looks like they're all questioned out.
All right.
Okay. Thank you. Whoops. Here's one. Thank you both for all you do. Okay. And
well,
thank you, Dr. Spencer, for sharing your research with us, and best of luck with it.
Thank you so much, and thank you for your time and support. I really appreciate the great questions and the great ideas. So thank you.
Yeah. And thank you to all the attendees who came and attended. I hope you all learned something, because I definitely also learned a lot.
Yeah, great.
Great.
Super.
Good.
All right.
Okay.
Thank you so much.
Thank you.